Hello guys. Today we're going to discuss chapter Principles of Inheritance. First of all, okay guys. Hi. So today we will be discussing PYQs from the chapter Principles of Inheritance. Um, so I have taken almost 90 questions from from this chapter. It will be covering NEET and related exams. Okay. So I will be showing questions here one by one. You can answer in the chat box. Then if you have any doubts, you can ask me. Okay. Fine. So let's start. Fine, guys. Let me take the first of all, we will be taking questions from Mendel's law of inheritance and inheritance of one gene. We will be covering topic by topic. Okay. Let me take the first question. The first question is the mother has A blood group, father has B blood group, and the child is A plus. What can be the possible genotypes of all three respectively? IA, IA, IB, IB small I. Choose the correct answer from the options given below. So what will be the answer here? Right? So concept we will be discussing as per the context. Okay. If you need more, you can let me know. Fine. So here they have mentioned that the mother is, here they have mentioned that, let me take a color. Yes. Here they have mentioned that the mother is A plus blood group and the father has B plus blood group and the child is A plus. What can be the possible genotypes of all three? So if the child is A plus, you know that if the child is A plus, there are only two options are possible. Either IA or IA small I, right? So this IB small I is not possible. This is not possible. So these three are possible. Now, here they have mentioned that the father is A plus. Father is A plus. Sorry, the mother is A plus and the father is B plus and the child is A plus. So what will be the answer here? When you take here, let's say for example, when you take this option, here IA small IA small I. So one is A blood group, another one is what? B blood group. Now when you take here IA small I, so here one small I, at least it should come from at least one parent now, but none of the parent do not have small I, so this won't be the answer. When you take here, you can see that IA small I, A blood group, B blood group, A blood group. Right here, IA small I is possible. If this one and this one cross, we will get IA small I. So B is possible as well as E is possible. Answer will be option D. Right? Yeah, very good. Option D is the answer. Nitya, correct. I hope it's clear. Okay. Let's move to the next question. In the next question, in a plant, black seed color, capital B, capital B, capital B, small b is dominant over the white seed color. In order to find out the genotype of black seed plant, in which of the following genotype will you cross it? Very nice question. You know that how can we find the genotype? Let's say when I say black, there are two probability. Either it could be either the genotype, either it could be capital B, or it could be capital B, small b. How can we determine that? We can determine it by doing test cross. Test cross means we can cross with a homozygous recessive. Yes or no? So answer will be what? Option D. And this cross is called test cross. Yes, very good, guys. Chat. Now, I hope it's clear. Now let me take the next question. A pink flowered snapdragon was crossed with a red flowered snapdragon plant. What type of phenotypes are expected in the progeny? Only pink flowered plant, red, pink as well as white flowered plants, only red flowered plant, red flowered as well as white flowered plant. So what will be the answer? Here they have mentioned pink snapdragon. Snapdragon means they are showing pink color because of the incomplete dominance. So here pink flowered plant, capital R, small r, is crossed with a red flowered plant, which means capital R, capital R. What type of phenotypes are expected? So this is pink and this one will be red. So what are possible here, guys? Capital R, capital R, then capital R, capital R, then capital R, small r, then capital R, small r. Yeah. So these two will be what? Red. These two will be pink. So what will be the option one? Only pink, which is not possible. Red, pink as well as white. White is not here. So it is incorrect. Only red. No. Red as well as pink. Yes. So option D is the answer. Very good. Very good. Now let me take the next question. Match list one with list two, right? In list one, you can see that two or more alternative forms of a gene, cross of F1 progeny with the homozygous recessive parent, cross of F1 progeny with any of the parent, number of chromosomes sets in a plant. Here list two back cross, ploidy, allele, and test cross. So what will be the answer here? You know, two or more alternative forms of a gene which indicate allele, right? A means allele. You see A3. Yes, in first option itself A3. None of the remaining do not have A3. So option 1 will be the answer. But we just need to confirm it. How can we confirm it? Right? A3, B4. Cross of F1 progeny with a homozygous recessive parent. Yes, this is called a test cross. True. Now C1. Cross of F1 progeny with any of the parent, which is called a back cross. Very good. Correct. Correct. So crossing of F1 with any of the parent, which is called a back cross. And if the parent is homozygous recessive, then the cross is called test cross. So we can say that test cross is a type of back cross. Now, number of chromosome sets in a plant. This is nothing but ploidy. So D2. So answer will be option 2. This was a question from NEET 2024. Yes, easy one. Now, let me take next question. Which of the following can be explained on the basis of Mendel's laws of dominance? Out of one pair of factor, one is dominant and the other is recessive. Alleles do not show any expression and both the characters are appears as such in the F2 generation. Factor occur in pairs in normal diploid plants. The discrete unit controlling a particular character is called a factor. Now, the expression of only one of the parental characters is found in a monohybrid cross. So, what will be the answer here, guys? Out of one pair of factor, one is dominant and other one is recessive. Alleles do not show any expression and both the characters are appears as such in the F2 generation. Factor occur in pairs in normal diploid plants. The discrete unit controlling particular character is called factor. The expression of only one of the parental characters is found in a monohybrid cross. So what will be the answer here? What do you guys think? So many are saying D, A, C, D, E. So in our NCRT, they have mentioned three laws, right? One is called a law of dominance. Another one is called a law of segregation. and third one is called law of independent assortment. So under law of dominance, they have given, some points. One of the point is what they have mentioned, that factors occur in pairs, which they have mentioned. Then also they have mentioned that out of a pair of allele, one will be dominant over the other. So here you can see A is correct. Now B is not correct. Alleles do not show any expression. Both the characters appears as such. In this is not related to law of dominance, right? Now uh, C is correct, D is correct, even E is also correct. Expression of only one of the parental characters is found in monohybrid cross, right? Because this F1 shows only one of the parent. Capital R with small r, small r. What we are getting? Capital R, small r, right? Which means, let's say if it is round, only one of the parental characters, right? So ACD, very good guys. Many are answering. Now, next question. As per ABO blood grouping system, the blood group of father is B plus. I think, do we done this? Yeah, I think we already done this. Yes or no? First question was this now. Um, no, this is I think different. As per ABO blood grouping system, the blood group of father is B plus, mother is A plus, and the child is O plus. The respective genotype can be, guys, similar type of question, right? What will be the answer here? Choose the most appropriate answer. So here they have mentioned child is O blood group, mother A, father B. If child is O blood group, then it will be small I, right? O means small I. Yes, this is true. This is true. This is not true. This is not true. This is not true. Right? Now, um, B blood group mean IBI, A blood group mean IAI. If small I comes, yeah, O we will get. This is fine. Here there is a problem. Out of this small I, if we need to get small I, at least one small I should be present in father and mother, na? So because of that, these are incorrect. What about this one? Yeah. So only A will be possible, na? Very good. Yeah. So only C will be the answer. This is not possible because this is missing. So C. Yeah. See, now you have seen a same type of question repeated in NEET exam. Okay. Now, a heterozygous pea plant with the violet flowers was crossed with a homozygous pea plant with the white flower. Violet is dominant over white. Which one of the following represent the expected combination among 40 progenies formed? 2023 Manipur question. Let's see. Heterozygous pea plant, which mean having violet flower. So here let me take capital V as violet, small v as white, because they have mentioned violet is the dominant, right? So over white. So heterozygous violet means what? Capital V, small v, crossed with a homozygous pea plant with the white flower, which means small v, right? Now, which one of the following represent the expected combination among 40 progenies formed? We will cross it, right? So this is violet, this is white. Now, capital V, small v, capital V, small v, small v, small v, small v, small v, right? What this will be? This will be violet. What it will be? Violet. What it will be? White. This also will be white, which means you can see what is the ratio between violet and white? 50-50 or 1 is to 1. So if there are 40, 20 will be from, I mean 50 percentage, okay? So 20 will be violet, remaining 20 will be white. So option B is the answer. this won't be, this won't be, this won't be. Yes, guys, very good. Everyone is answering. Now let me take the eighth question. Given below are two statements. Statement one, the Mendel studied 7 pairs of contrasting traits in pea plants and proposed law of inheritance. Statement 2, 7 characters examined by Mendel in his experiment on pea plant were seed shapes, color, flower color, pod shape, pod color, flower position, and stem height. In the light of above statement, choose the correct answer. What will be the correct answer here, guys? Mendel studied 7 pairs of contrasting traits, which is true. 7 pairs of contrasting traits, right? Here you need to be aware if they are asking pairs of contrasting trait or contrasting pairs, then the answer will be 7. But if they are asking true breeding, then the answer will be 14. I will be discussing later. Okay. Now, in statement 2, 7 characters examined by Mendel in his experiment on pea plants were seed shape, true, seed color, flower color, correct only, pod shape, pod color, flower position is correct, stem height also correct because tall or dwarf, all are correct only. So, both statement 1 and 2 are correct. Right? Very good, guys. Very good. Now, ninth question. The production of gametes by parents, formation of zygote, the F1 and F2 plants can be understood from a diagram called punch square, Punnett square, net square, bullet square. Very easy. What is the answer? Punnett square. Right? Very good. Yes, Nitya, correct. Next, 10th one. Identify the wrong statement with a reference to the gene I that controls ABO blood groups. What is the wrong statement? A person will have only 2 of the 3 alleles. When IA and IB are present together, they express same type of sugar. Allele I does not produce any sugar. The gene I has 3 alleles. What will be the answer? A person will have only 2 of the 3 alleles. True. For example, if my blood group is AB blood group, I will have only 2 alleles, which is IA and IB. Let's say here Nitya's blood group is O blood group, which means small I, O blood group. Now, if I take me as well as Nitya as a population, totally how many alleles are there? IA, IB, and small I, which means this is called a multiple allelism. So, multiple allelism present in population, not present in a single individual. If you take a single individual, only 2 alleles will be there, right? Yes. When IA and IB present together, they express same type of sugar, which is incorrect. If I take IA, IB, I will get AB blood group, not same type. Allele I does not produce any sugar. True. Gene I has 3 alleles, which is true because there will be 3 alleles. IA, IB, and small I. Right? No, B is not correct. When I and IB present together, they express same type of sugar. No, they are not expressing same type now. They are expressing different type. One is A, another one is B. That's why it is incorrect. So answer will be option B. Clear? Yeah, very good, guys. 11th one. How many true breeding plant varieties did Mendel select as pairs, which were similar except in one character with contrasting traits? What will be the answer here? They're asking how many true breeding pea plant variety. If they're asking true breeding, always remember answer will be 14. If the true breeding, answer will be 14 because uh, if let's say for example tall, dwarf, yellow seed color, green seed color, etc., etc. So 14. Now another twist. Number of contrasting characters studied by Mendel for his experiment was 4, 2, 7, 14. Here, guys, answer will be 7. If they ask contrasting characters, answer is 7. But if they ask true breeding varieties, then answer will be 14. Always remember. Both asked in NEET exam. Clear? Yes. Now, 13. In Antherinum's snapdragon, the red flower was crossed with a white flower in F1 generation. Flowers were obtained. In F1 generation, pink flowers were obtained. When pink flower were self, F2 generation showed white, red, and pink flowers. Choose the incorrect statement from the following. This experiment, yeah. So, what will be the answer here? So here what happens in Antherinum? Antherinum shows incomplete dominance. Red flower, which means capital R, crossed with white, which means small r. And F1 shows pink, which means capital R, small r. This is pink and it is F1. Now they are selfed. In F2 generation, white, red, and pink were obtained. Choose the incorrect statement from the following. This experiment does not follow the principle of dominance, which is true because this experiment shows incomplete dominance. Pink flower in F1 is due to incomplete dominance. True. Ratio of F2 is 1 is to 2 is to 1, which is true now. If you are crossing, you will get it. Capital R, small r with the capital R, small r. Capital R, capital R, capital R, small r, capital R, small r, and small r, small r. This is red, this is pink, this is white. So what is the ratio? 1 is to 2 is to 1. 1 is red, 2 is pink, 2 are pink, and 1 is white. This is also true. I think they're asking incorrect now. I think one option is missing here. One option is missing here. That will be the answer. Okay. B is correct only. Pink color in F1 is due to incomplete dominance. Yeah, that is true. So D will be the answer. Okay. Now, question number 14. Yeah, all correct, but I think option D is missing here. And D will be the answer because that is the incorrect statement. Now, question 14. Here, give me 1 minute, guys. Yes, let's continue. Question 14. Which of the following characteristics represent inheritance of blood groups in humans? Dominance, codominance, multiple alleles, incomplete dominance, and polygenic inheritance. So, what will be the answer here? Dominance is correct. Codominance is correct. For example, IA, IB, they show codominance. Both are expressing. Dominance is correct because when IA small I comes, only A is present. So dominance is correct. Multiple alleles, true, because 3 alleles are here, IA, IB, and small I. So multiple alleles, which is also true. But incomplete dominance is not true. Polygenic inheritance also absent in case of AB or blood grouping, right? So A, B, C will be the answer. What is A, B, C? Option B. Yeah, very good, guys. Kausalya, yeah, Nitya, Raghotama. Oh, very nice, guys. Nice. 15. Among the following characters, which one was not considered by Mendel in his experiment on P? Stem, tall or dwarf. Trichomes, glandular or non-glandular. Seed, green or yellow. Pod, inflated or constricted. What will be the answer here? Stem, tall or dwarf, which is true, right? It is true. Trichomes not considered in Mendel's experiment, right? Yeah, seed, green or yellow. True. Pod, correct. B is the incorrect statement. So B will be the answer. Very good. 16. The genotypes of husband and wife are IA, IB, and IA small I. Among the blood types of their children, how many different genotypes and phenotypes are possible? Let me try. So which mean, let's say husband is IA, IB, wife IA, small I. Let's cross it. IA, IA. Another cross. IA, small I. Now another cross. IA, IB. And another cross. IB, small I. Correct now? So this is, these two represent A blood group. This represent AB blood group. This represent B blood group. Now you tell me guys, how many genotypes are there? One, two, three, four. There are four genotypes, right? How many phenotypes are there? A, B, B. Three phenotypes. So what will be the answer? You tell me. Option C. C will be the answer now. Yeah, very good guys, very good. Now, which one from those given below is the period for Mendel's hybridization experiment? 1856 to 1863 means the Mendel conducted experiments for 7 years, right? Option A. Now, a true breeding plan is nearly homozygous and produce offspring of its own kind, always homozygous, recessive in its genetic constitution, one that is able to breed on its own, produced due to cross pollination among unrelated plants. Guys, what is true breeding? For example, capital A, small a, all these are true breeding. Capital A, small a is not a true breeding one. So they are homozygous and produce offspring of its kind. True, right? This can produce capital A, this can produce small a, right? Or fine. Always homozygous recessive, which means they are saying it will be always small a. That is not correct because it can be capital A, or homozygous dominant as well, that which is also possible. So B is wrong. One that is able to breed on its own, no. Produced due to cross pollinate, no. True breeding will be given by or will be produced by self-pollination, not cross-pollination. That's why we say continuous self-pollination will be giving to true breeding. But if it became over, which will be leading to a condition called inbreeding depression, right? Yes. So option A will be the answer. Next. In a test cruise involving F1 dihybrid flies, more parental type offspring were produced than recombinant. This indicate the two genes are located on two different chromosomes. Chromosome failed to separate during meiosis. The two genes are linked and parent and present on the same chromosome. Both of the characters are controlled by more than one gene. So in a test cross, F1 dihybrid play more parental compared to recombinant type. If there are more parental, what it indicate? See, if more parental present, it indicating that linkage is yeah. You can remember like this. Linkage is directly proportional to parental parental type parental type offsprings, which is inversely proportional to recombinant types. Recombinance. So here they have mentioned more parental. If there are more parental, indicating that two genes are linked. For example, if you take a one chromosome, if there are two chromosomes, if there are two genes A and B, if they present together, which means there is a linkage between them. If there is linkage, then parental type will be more, recombinant will be less. So here answer will be option C. Two genes are located on two different chromosomes, which means in that case there won't be any linkage. Cancel. This won't be the answer. This won't be the answer. So option C is the answer. I hope it's clear. Now, 20. A tall, true breeding garden pea plant is crossed with a dwarf, true breeding garden pea plant. When the F1 plants were self, the resulting genotypes were in the ratio of, let me try, tall, true breeding, which means capital T, crossed with dwarf true breeding, which means small t. And what we will be getting in F1, we will be getting capital T, small t. This will be the F1. Now they are saying they are selfed. Selfed means what? Capital T, small t with the capital T, small t. What we will be getting? Capital T, capital T, capital T, small t, capital T, small t. Now small t, right? Both of this will be tall. All these three will be tall. This one will be what? Dwarf. So what is the uh how many are tall homos? Tall homozygous one. Tall homozygous one. Yeah, true. Only this is tall homozygous one. Two are tall heterozygous two. Yeah, these are two heterozygous and they are tall. One is dwarf one. So this is true. This is incorrect. Tall heterozygous one. No, which is two. So answer will be option A. Very good, guys. Nitya, Padma, everyone, nice, nice, nice. Let's move to the next. How many pairs of contrasting traits, contrasting characters in pea plants were studied by Mendel in his experiment? Guys, repeated question. What will be the answer? How many pairs of contrasting characters? If contrasting characters comes, what will be the answer? Seven. So same type of question with a slight twist repeated three times. Okay. Yeah. This is the important of PYQs. Question will be around this topic, modification only. Okay. Now, a man with a blood group A marries a woman with a blood group B. What all are all possible blood groups of their offsprings or children? Yes. A man with a blood group A marries a woman with blood group B. Okay. In this case, if here they have, when I say A blood group, there are two probability. It could be IA or IA small I. Since they haven't mentioned anything, we can go with IA small I because this gives maximum variety. We will be going with unless and until they are not mentioning anything. If they have specifically mentioned homozygous, then we will go with this. Otherwise, we will go with this only. Okay. I am taking IB small I. Now let's cross IA, IB. Then IA small I. Then IB small I. Now small I. Guys, tell me what is this? A blood group, A blood group, B blood group, O blood group. So what are possible? Sorry, this is AB blood group, IA, IB. So all four are possible. A, B, A, B, O. True. No, no, no. Answer will be option one. Yeah, very good. Very good, guys. All are correct. Yeah. Now, 23. Multiple alleles are present in at the same locus of the chromosome on non-sister chromatids on different chromosome at different loci on the same chromosome. Guys, what will be the answer? When you take a chromosome, you can see, let's say, capital A, small a, which means this is same locus. Option A will be the answer. Clear? Now, 24. Alleles are different molecular forms of a gene, heterozygotes, different phenotype, true breeding homozygotes. Guys, what will be the answer? Nothing but option A, right? Alleles are different molecular forms of a gene, not heterozygous. It can be homozygous also, no problem. Yeah, very good. A gene showing codominance has alleles tightly linked on the same chromosome Alleles that is recessive to each other, both alleles independently expressed in heterozygote, one allele dominant on the other. What will be the answer? This won't be answer. If it is one allele dominant on the other, which is this is called a dominance. This is called a dominance. Here it is codominance. So this won't be the answer. Both alleles independently expressed in heterozygous. This is true because when we take IA, IB, you see both are expressing now. That's why we are getting A and B, AB blood group. So this is true. Alleles that is recessive to each other. No. Alleles tightly linked. No. So answer will be option C. Yeah, Nitya, correct. Option C. Now, in his classic experiments on pea plants, Mendel did not use, guys, underline, did not use pot length, seed shape, flower position, and seed color. What will be the answer? He used seed shape, which means round, wrinkled. He used seed color, which means yellow and green. Flower position, yes, axial, terminal. Pot length, no. What they have used? They used pot shape, not length, guys. They use pot shape as well as pot color, right? Not length. So answer will be option. So what will be the answer here? Option A. Clear? Very good, guys. Harshavardhan, nice, nice. Now, 27. If two persons with the AB blood group marry and have sufficiently large number of children, these children could be classified as A blood group, AB blood group, and B blood group in 1:2:1 ratio. Modern technique of protein electrophoresis reveals the presence of both A and B type of protein in AB blood group individual. This is an example of, guys, this question looks like a big question. Actually, it is very, very simple. Whenever you see very long question, imagine mostly that will be easy question. So here simply they have given a big story. Here nothing but if you know both A and B are visible in an AB blood group, what it is called? This is called codominance. For example, when we take IA, IB, both A and B is visible. This is called what? Codominance, right? Yeah, both A and type of protein is available. AB blood group individuals show codominance, not complete dominance, not incomplete, not partial. Yes, very good. B only, Nitya. Correct, correct. Kausilya, Anud, Harshavardhan. Nice guys, very nice. Now, 28. Which Mendelian idea is depicted by a cross in which the F1 generation resemble both the parents? Codominance, incomplete dominance, law of dominance, inheritance of one gene. What will be the answer, guys? Crows in which F1 generation resembles both parents. What it will be? For example, if you are crossing, let's say, how can I say? In case of cattles, let's say you are taking male part is male partner is, let's say, black, female partner is white, then you know what will be the progeny will be black and white, or let's say black cattle with the white patches. This is showing both now. This is called codominance, guys. Codominance. Now you know why not incomplete dominant. Many people may think incomplete dominant. In incomplete dominance, when we are crossing red and white, we are getting pink. This is red. This is white. What we are getting? We are getting pink, which means in F1 do not have both color. F1 is having an average color. You know, pink is an average color between red and red and white. But here, what is the difference? In F1 progeny, both black and white comes. This is called a codominance. Here, what is the difference? Here you see pink. Both of the parent do not have pink, which means they are getting a color in between. Yeah, very good. Nitya, very good. Intermediate. This is called incomplete dominance. Incomplete, sorry, incomplete dominance. This is called incomplete dominance. So what will be the answer? Option A, right? Very good, guys. Very good. Now, now we are entering into the next topic called inheritance of two genes. I hope you guys are ready for it, right? Let's see. When a tall pea plant with a round seed was selfed, it produced the progeny of. Let's see. A tall pea plant with a round seed. Huh. What are the statement they have given? Tall plant with a round seed, tall plant with a ringle seed. Identify the genotype of the parent plant. Okay, okay. Um, yeah. Here they have mentioned tall as well as round, then self. We don't know their genotype, but we are getting tall and round seed, tall and ringle seed. If that is the case, um, how can we do this? Identify the genotype of the parent plant. We need to guess then and later we need to confirm it. How can I do it? Let me guess it like, hmm, when I say tall, there are two options. Capital T or capital T small t. When we say round, there are two probabilities. Capital R or capital R small r. So here, since we are getting both round and ringle seed, one partner I am taking it as capital R small r. And here only tall is coming now. So I am assuming, it's my just guess. I am assuming the tall I am taking as capital T instead of capital T small t. This is my guess only, but I will do it and I will verify it. Here you see what will happen. Capital T from this parent. Here there will be two gametes. If we cross capital T, capital R, capital T, small r. So what is this capital T, capital R means what? Tall, round. Tall, round seed. Here, tall, small r means what? Wrinkled seed. Tall, round seed. Tall, wrinkled seed. Both are correct. So answer will be option D. I am lucky. My guess was correct. Suppose, imagine you don't know how to guess. Then only option you have to do everything. I will take this. Let me try this one. If I am doing this, capital T, capital T, capital R with the capital T, capital T, capital R, capital R. You see what we will, all children will be tall and round. We will not get this one. B we won't get. So this won't be the answer. Now let me take this one. Capital T, small t, capital R with the capital T, small t, capital R. So here what are possible? Capital T, yeah, capital T, capital R, small t, capital R. Here, capital T, capital R, let, then small t, capital R. Now, if I cross, what will happen, guys? Capital T, capital T, capital R, capital R, capital T, small t, capital R, capital R, capital T, small t. Here, small t, capital R, capital R. See what we are getting? Tall, round. Here tall round, wrinkled round. Sorry, no, sorry, dwarf round. So all these are not working. If you cross, you will get this won't work. Only this will work. I think I will do this crossing again for you guys. I think there were some change. Yeah. So here this is one. Yeah. This is the selfing, guys. Like earlier I done only with the gametes. So what will possible here? Here there will be two types of gamete. Capital T, capital R, then capital T, small r. Here again capital T, capital R, capital T, small r. Then you cross. What will happen here? You see capital T, capital T, capital R, capital R. Another one capital T, capital T, capital R, small r. Then capital T, capital T, capital R, small r, capital T, capital T, small r, small r. Capital T, capital T, small r, small r. See, this is what? Tall, round. What is this? Tall, wrinkled. Tall, round, and tall, wrinkled. We are getting only these two now. I hope it's visible. Yeah. So this is the answer, right? Fine, guys. Fine. Let's move. I hope it's clear. Yeah. Next. Match list one with list two. List one, monohybrid cross, dihybrid cross, incomplete dominant test cross. 1:1, 1:2:1, 3:1, 9:3, 3:1. Guys, what will be the answer? Choose the correct answer. Monohybrid cross, 9:3:3:1 will be what? Dihybrid cross, right? B4, B4. Yeah, so we can remove C and D. Now, what else? Monohybrid cross, dihybrid cross. Test cross, test cross is 1 now, 1:1. D1, again D1. Yeah. Now you can see incomplete dominance means what? 1:2:1. So C2, C2, C2. Yeah. A3. Monohybrid cross 3. Option A will be the answer. You may have a question, right? Because when you take monohybrid cross, phenotypic ratio is 3:1, genotypic ratio is 1:2:1. Yes or no? So you may think, some people may think, why can't we connect monohybrid cross with the 1:2:1? But the point here, for incomplete dominance, their phenotypic, here they have mentioned here. Here it is phenotypic ratio. So for incomplete dominance, phenotypic ratio is what? 1:2:1. For monohybrid cross, phenotypic ratio is what? 3:1. Right? So option A will be the answer. Yeah. Kausalya, Padma, Nitya, Shanmati. Yeah, very nice, guys. Very nice. Now, 31. A certain plant homozygous for yellow seed and red flower was crossed with a plant homozygous for green seed and white flowers. The F1 plants had yellow seeds and pink flowers. The F1 plants were selfed to get F2 progeny. Assuming independent assortment of the two characters, how many phenotypic categories are expected for these characters in the F2 generation? Guys, this is a nice question with a nice twist. It's not our normal question. Because you see, here yellow seed and red flowers crossed with a plant homozygous for green seed and white flower, right? And the F1 plant had yellow seed and pink flower. If pink comes, if pink comes, indicate that the second gene shows incomplete dominance. We have to be careful here. So let me write yellow seed and red flower, which mean seed color na. So capital Y, capital Y, capital R with small y, small r. So yellow round crossed with the green white flower. No, no, some problem is there now. Yellow seed, red flower. Yellow seed, red flower. Yellow seed, red flower. Now you see, here we will be getting capital Y, capital R, small r. What are the characters? Yellow, pink, which mean here flower color shows what? This R gene shows what? Incomplete dominance. So normally we will be getting 9 is to 3 is to 3 is to 1, which means there will be 4 types. But here it is not applicable. Why? Because here there is incomplete dominance. So how to do this? That is a question, right? Guys, answer is different, guys. I will do this for you guys. Okay, let me do it. Uh, just note it down, guys. Here, what we need to here we are crossing yellow red with the green white. I will do it for you guys. Now, here, when you take yellow can be represented by capital Y, or capital Y, small y. Correct now? Yellow. Green can be represented by small y. Right? Yeah. This is one gene. This is seed color. Now let me take flower color. In flower color, red can be indicated by what? Or let me write. If I take capital R, if I take capital R, or capital R, small r, which indicate red. Now, if I take, sorry, only capital R represent red. If I take capital R, small r, it indicate what? Pink. If I take small r, it take white. Guys, I am giving you the route map for finding the answer. You can, if you just follow this, you will get the answer in NEET exam. Okay, now you see, when you take the flower color, there are 3 varieties. Now, what we need to do? We need to cross the seed with the flower. First, cross these 3. Let me write. What will be the first phenotype? Yellow seed. Yellow seed with the red flower. Red flower. What will be the second phenotype? Yellow seed with the pink flower. Yellow seed, pink flower. What will be the third type, guys? Yellow seed with the white flower. Yellow seed, white flower. Now another cross. What will be this cross? Cross green with the red flower, pink flower, as well as white flower. So what will be the fourth phenotype, guys? The fourth phenotype will be green seed with red flower. What will be the fifth one? Green seed with pink flower. What will be sixth type? Green seed with green seed with white flower. Now you see, guys, how many phenotypes are there? There are 6 phenotype. 1, 2, 3, 4, 5, 6. So what will be the answer? The answer will be option D. Option D will be the answer. Okay. I hope it's clear. Yeah, let's move to the next question, guys. What is the expected percentage of F2 progeny with yellow and inflated pod in dihabit cross experiment involving pea plants with a green colored inflated pod and yellow colored constricted pod? Let me do it. Expected percent of F2 progeny with yellow and inflated pod. Guys, in a dihybrid cross. Guys, pod, in pod color, what is the dominant? Green, dominant, yellow, recessive, right? Yeah. And similarly, in pod shape, which is dominant, inflated is dominant, and constricted shape is recessive, right? Recessive. Yeah. Now let me cross, guys. Let me cross. So here they have mentioned what is the expected percent F2 progeny with yellow and inflated pod in dihybrid cross experiment involving pea plant with what is the expected percentage of F2 progeny with yellow and inflated pod in dihybrid cross experiment involving pea plants with green colored inflated pod and yellow colored constricted pod. Here what will be the answer, guys? Pea plant green colored, pea plant green colored, which means let me write capital G, inflated, capital I with yellow, which means small g, small I. Yeah. Okay, crossing spin involving pea plant with a green colored inflated pod and yellow colored constricted pod. And they are asking what? Yellow and inflated. Yellow means what? So if we cross this, what will be getting? Capital G, capital I, small I, which mean green pod with the inflated. So here what they're asking, yellow and inflated. So there is a short way to answer this. Yellow is what? A recessive character. Inflated means what? Dominant character. Now you know that this is similar to capital R, capital Y, small y. Yes or no? Now, if you do this normal dihybrid cross, we will be getting phenotypic ratio as 9 is to 3 is to 3 is to 1. What is 9? Two dominant characters. What is 3? One dominant, one recessive. What is 3? One recessive, another dominant. What is 1? Two recessive characters. So here they are asking one recessive and one dominant now. One recessive and one dominant means what? They're asking this one, which means what is the ratio of this one? The ratio of this one will be 3 out of total 9 plus 3 plus 3 plus 1, 16 into, they're asking in percentage, right? Into 100. What will be the answer, guys? 18.75 percentage. Option D. Is it clear? Now, after some time, you can directly answer for it. If once you become an expert, you can directly answer for it, right? Yes. Now, 33. Fruit color in squash is an example of inhibitory genes, a recessive epistasis, dominant epistasis, complementary genes. Here answer will be dominant epistasis. Okay. Now, we are getting into the next level of questions, guys. Chromosomal theory of inheritance. Very important area. Okay. Now, here, how the questions come. The chromosomal theory of inheritance was proposed by Robert Brown, Morgan, Sutton, and Bovary, Mental. What will be the answer? Chromosomal theory of inheritance by Sutton and Bovary, right? Not Mendel, not Morgan, not Brown. 35. Experimental verification of chromosomal theory of inheritance was done by. Who done the experimental verification, guys? Experimental verification was done by Morgan. Option C is the answer, right? Morgan. 36. Chromosomal theory of inheritance was proposed by. Who proposed chromosomal theory of inheritance? Bateson, Punnett, Morgan, Watson and Crick, Sutton and Bovary. Yes, 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 guys. Sutton and Bovary. Sutton and Bovary. Right? Watson and Crick proposed, I remember, double helix model of DNA. Not this, right? 37, guys. What map unit is adopted in construction of a genetic map? Option A unit distance between two expressed genes representing 10% crossover. Option B, a unit of distance between two expressed genes representing 100% crossover. Option C, a unit of distance between genes on chromosome representing 1% crossover. Then option D, a unit of distance between genes on chromosome representing 1% crossover. Guys, you know that one map unit will be equal to 1% of recombination. 1% of recombination. Similarly, 10 map 10 map unit will be equal to 10% of recombination, right? I mean 10% crossover. So what will be the answer here, guys? 1% crossover. Yes, Harshavardhan. Yeah, yes. Yes, guys. Option C is the answer. Now we are getting into the questions related to linkage and recombination as well as polygenic inheritance and pleiotropy. Important topic, guys, right? Now, what will be the answer here? Given below are two statements. Statement one, when many alleles of a single gene governs a character, it is called polygenic inheritance. In polygenic inheritance, the effect of each allele is additive. What will be the answer, guys? When many alleles govern a single gene, many alleles of a single genes govern a character, it is called polygenic. It is wrong, guys, because polygenes mean many genes, not single gene. Hence, it is wrong. In polygenic inheritance, each allele is additive. That is true. For example, when you take a small a, small b, small c. In case of skin color, these are albinos. But if you take capital A, small a, capital B, small b, capital C, small c, here there are three dominant alleles. So here, you know, the color will be more. Let's say if you take capital A, capital B, capital B, capital C, small c. Let's say here, how many dominant allele? One, two, three, four, five. There are five dominant alleles. So here, skin pigmentation will be more dark compared to this case because here only three are there, which means if number of dominant alleles increases, automatically the phenotypic expression also will be changing. That's why each allele have additive effect. Hence, option statement 1 is false, 2 is true. Option B is the answer. Yes, very good, very good. Yeah, Harshavardhan, Padma Priya, nice guys. Now, 39. Match the following. List 1 and list 2. Type of inheritance, incomplete dominance, codominance, pleiotropy, polygenic inheritance. List 2. Blood groups in humans, flower color in antherinum, skin color in humans, and phenylketonuria. How can we connect, guys? Skin color in human is polygenic inheritance. T3. Which option 1 gone? Option this gone, this gone. Yes, guys, option 2 will be the answer. Right? We are lucky. Now we just need to confirm by cross-checking. A2, incomplete dominance, flower color in antherinum. True. B1, codominance, blood groups in human. True. C for pleiotropy, phenylketonuria. So answer will be option B. Yes, yes, very good, very good. Now guys, 30, 40. The phenomenon of pleiotropism refers to more than two genes affecting a single character, presence of several alleles of a single gene controlling a single crossover, presence of a two alleles, each of the two genes controlling a single trait, a single gene affecting multiple phenotypic expression. Is what is pleiotropy? Pleiotropy means, always remember, pleiotropy means multiple phenotypic expression. Single gene responsible for multiple phenotypic expression. So more than two genes? No. Single gene responsible for multiple phenotypic expression. This is called pleiotropy. So more than two genes, wrong. Several alleles of a single gene controlling single crossover. No. Presence of two alleles, each of the two genes controlling a no. Single gene affecting multiple phenotypic expression. Option D will be the answer. Right? Yes, guys. Very good. Very good. Now, 41. The frequency of recombination between gene pairs on same chromosome as a measure of the distance between genes to map their positions on chromosome was used for the first time by Henking, Thomas Hunt Morgan, Sutton and Bowery, Alfred Stotwan. Guys, this is nothing but what? Gene map, right? For gene map, the distance between genes, the distance is measured by recombination frequency. This method was developed by Alfred Stotwan. Alfred Stotwan was student of the Morgan. He was Morgan students. Okay, fine. 42. Given below are two statements. One is labeled as assertion and another one is reason. What is assertion? Mendel's law of independent assortment hold good does not hold good for the genes that are located closely on the same chromosome. Reason: closely located genes assort independently. Is what will be the answer here? In case of Mendel law of independent assortment, not hold good for the genes that are located closely on the same chromosome. This is true, right? Because if in a chromosome, if there are two genes like A and B, if they are closely present, which indicates that there is a physical association between them called linkage. If linkage is present, independent assortment will be absent. No independent assortment. No independent assortment. Hence, this statement is true. Closely located genes assort independently. This is wrong, because if they are closely located genes, then they shows linkage. If they shows linkage, then independent assortment is not possible, right? So what will be the answer? Assertion is correct, but reason is not correct. Option D. Yes, guys, very good, very good. Let's move to the next question. The best example of pleiotropy is phenylketonuria, color blindness, ABO blood group, skin color. Pleiotropy, what will be the answer, guys? Phenylketonuria, right? ABO blood group shows codominance. Color blindness, not skin color, not skin color is shown by polygenic inheritance. Or skin color is an example for polygenic inheritance. Very good, very good. 44. The frequency of recombination between gene pairs on the same chromosome as a measure of the distance between genes was explained by Morgan, Mendel, Alfred Stotwan, Sutton and Bowery. Guys, what is the answer? Here answer will be what? The frequency of recombination as distance between genes. Proposed by Alfred Stotwan. Option C will be the answer. We already discussed it now. Repeated. Question is repeated. Now, select the correct statement. Franklin Stahel coined the term linkage. Punnett square was developed by British scientists. Spliceostom takes part in translation. Transduction was discovered by Altman. Guys, what is the correct statement here? The first one is not correct, because linkage is related to Morgan, right? Then Punnett square was developed by a British scientist, which is true. And what is the name of that scientist? The name of the scientist was Reginald C. Punnett. Reginald C. Punnett. Spliceosome takes part in translation, which is wrong because spliceosomes are present in splicing. And splicing is a part of post transcriptional modification. It is a part of uh, post transcriptional modification. It is not a part of translation. It's not a part of translation. Hence, it is wrong. Transduction, no, this is also wrong. So, answer will be option B. Clear? Very good, guys. When you are discussing PYQs, see, same question may not come, but related question comes. So, when you are working on a question, you have to think how they can formulate another type of question from here. If you think in that way and study, you will crack NEET. Okay. Yeah. Match column 1 with the column 2 and choose the correct option. Column 1, dominance, codominance, pleiotropy, polygenic inheritance. Yeah, here correct option now. Yeah. Column 2, many genes govern a single character. Then in a heterozygous organism, one allele express itself. Then in a heterozygous organism, both alleles express themselves fully. Then a single gene influence many characters. Guys, what will be the answer here? Many genes governs a character. Many genes means what? Many polygenes, right? Polygenic inheritance. D1. D1. Yes, we got the answer. We are lucky, right? D1. Now you see A2. Dominance. Yeah, heterozygous organism, one allele express itself. That is called the dominance. Yeah, B3. Codominance. In heterozygous organism, both alleles are expressing. Yeah, C4. Pleiotropy, a single gene responsible for or single gene influence many characters. All are correct. Option B is the answer. Very good, guys. Very good. Very good. Now, let me take 47. The movement of a gene from one lineage group to another is called. The movement of gene is called what? Translocation, not inversion, not crossing over, not duplication, right? Very good. Then the term linkage was coined by Bowery, Mental, Sutton, Morgan. Guys, linkage was, whenever the question comes related to linkage, go with Morgan, T.H. Morgan, okay? Right? So here answer will be Morgan. Now, go with the next question, 49. A pleiotropic gene is a gene evolved during Pliocene, control a trait only in combination with another gene, control multiple traits in an individual, is expressed only in primitive plants. Primitive plants. Pleiotropy means what? Always remember single gene influence multiple phenotypic expression. So what will be the answer? Control multiple traits. Yes, a single gene control multiple traits, which is called pleiotropic gene. C is the answer. Now let's take another question for NEET 2013. Which of the following statement is not true for two genes that show 50% recombination frequency? Guys, here, if the genes are present on the same chromosome, they undergo more than one crossover in every meiosis. The genes may be on different chromosomes. The genes are tightly linked. The genes show independent assortment. Nice question, guys. What will be the answer here? Maybe you may be knowing the answer, but do you know the explanation? That's a question. Do you know why other options are not correct? Let me ask you one thing. If a gene shows 50% recombination frequency, if two genes shows 50% recombination frequency, are they present on same chromosome or on different chromosome? What do you think? The thing is, they could be Both are possible. They could be either same chromosome as well as different chromosome. Let's say, um, this is a chromosome, and if they show 50% recombination frequency, genes are present on same chromosome and they undergo more than one crossover in every meiosis. This is true. Now, the genes may be different chromosome. True, because yeah, that's why I said genes could be either same chromosome or different chromosome. Both are possible in terms of, in case of 50% recombination frequency, right? But genes are tightly linked. This is not possible. Let me take. There is a chromosome. I am taking two genes, A gene and B gene. Here you can see A and B genes are closely present together. They are, the distance between them is very small, which means this linkage is tight linkage. In case of tight linkage, 50% recombination frequency is not possible. In case of tight linkage, all will be parental, recombinant will be zero. So as much two genes are coming nearby, parental will be increasing because of linkage, and recombinance or recombination frequency will be decreasing. Okay, so that's why C is wrong. Genes show independent assortment, which is wrong. Sorry, this is true because they are not linked, right? Yeah, option C will be the answer. Now guys, we are entering into another easy area called sex determination and mutation. See, the importance of sex determination is that from here you will get easy question, and sex determination is one of the favorite part for the it is favorite part for the people who create the question for NEET exam. You see, there will be question from this area, and it's not a that much tough part. It's easy only. Okay, we'll see. Now select the correct statements with respect to mechanism of sex determination in grasshopper. It is an example of female heterogamety. Male produces two different types of gametes either with or without X chromosome. Total number of chromosomes, which is autosomes and sex chromosome, is same in both male and female. All egg bear an additional X chromosome besides the autosomes. Yes, what will be the answer here? Here you can see a sex determination. It is an example of female heterogamety. In case of grasshopper, their sex determination will be XXXO type. XX will be female, XO will be male. So it is an example for female heterogamety. No, this is an example for what? Male heterogamety. Male is heterogamety here. So hence it is wrong. Male produces two different types of gametes either with or without of X chromosome, which is true. Male can produce two types, one gamete with X, another gamete without X. True. Total number of chromosome, which is autosomes and sex chromosome, is same in both males and female. No guys, here female has two X chromosome, but in male have only one X chromosome, which means male contain less number of chromosome when comparing with the female, right? Hence, this is wrong. All eggs bear an additional X chromosome besides the autosome. True, right? Because when you take, this can form two types of gametes, guys. Both having X chromosome. Both having X means what? One autosome plus X chromosome. Then this means what? Autosome plus X chromosome, right? So in this way, this is also correct. So what will be the answer? BD. BD, yeah, option A will be the answer. Clear? Yes, yes, Padma Priya. Male heterogametry. Very good. Now, guys, 52. Select the incorrect statement. Male fruit fly is heterogametric. In male grasshoppers, 50% of sperms have no sex chromosome. Then, in domesticated fowl, sex of progeny depends on the type of sperm rather than egg. Then, human males have one of their sex chromosome much shorter than the other. So what will be the answer here? Male fruit fly is heterogametic. So when you are coming in terms of fruit fly shows XX XY mechanism. Here XX will be female, XY will be male. So male is heterogametic, true. Which means this statement is true. Now, in male grasshoppers, 50% sperms have no sex chromosome. In male grasshopper means what? XO type. They produce two types of chromosome, two types of gametes. 50 percentage do not have sex chromosome. Hence, this is true. In domesticated fowls, sex of progeny depends on the type of sperm rather than egg. You know, when you take fowls, they shows what kind of sex determination. They shows ZZ ZW type, right? Here, ZZ will be male, ZW will be female. So they shows female heterogamety. So in domesticated fowl, sex of progeny. So here you see female produce two types of gamete, Z and W types. So here sex of progeny will be dependent on sperm rather than, sorry, this, so here sex of progeny will be dependent on whom? Sex of progeny will be dependent on egg, not sperm. Here they have given in reverse manner. Hence it is wrong. Sex of progeny depends on what? Depends on egg, not sperm, right? Hence it is wrong. Human males have one of their sex chromosome much shorter than the other. That is true because human male shows what? XY type. You know that in case of XY, Y is shorter than the X. So hence this also wrong. So what will be the, oh my God. Yeah, this is true. It is true. So what will be the correct answer? Option C, because this is the wrong statement, right? Very good. Now, next one. Here you can see that which of the following pairs is wrongly matched. Star synthesis in P, multiple alleles, ABO blood grouping, codominance, exotype of sex determination grasshopper, TH Morgan linkage. What will be the answer, guys? Wrongly matched. Here, star synthesis in P, multiple. This is wrong because nothing to do with multiple alleles. ABO blood grouping codominant, true, because AB blood group IAIP shows codominance, true. Exotype of sex determination grasshopper, true. Morgan linkage, true. So what is the wrong? Option A, right? Now 54, guys. An abnormal human baby with a triplex sex chromosome was born to fusion of two ova and one sperm, fusion of two sperms and one ovum, formation of abnormal sperms in the father, formation of abnormal ova in the mother. See, in this case, what do you need to do? What is happening is that, let's say, imagine there is a female with a triplex chromosome, and uh, she's marrying with a normal human, which is XY, normal male. So here they could produce one gamete with the 2X chromosome. The lady may produce one gamete with the 2X chromosome. From here, let's say uh, let's say this male is producing one gamete with the X chromosome. Now, if these two fuses, what we will be getting? Triplex. Triplex means what, guys? Yeah. This is what? Abnormal human baby with a triplex sex chromosome was born to whom? So what will be the answer here? Formation of abnormal ova in mother. You see? Abnormal ova in mother. Correct. Abnormal sperm not. Fusion of two sperms not. So answer will be option D. Yes, very good, guys. Very good. Right? Now, we have completed 54 questions. And you know that this is a big chapter. And this chapter carrying 10 percentage marks of the whole exam, right? You may get 5 to 6 questions from here. which mean 20 to 25 questions, right? 20 to 25 marks, which is a big thing. Even 1 mark can create too many changes in your ranks. So we have almost 50 more question, because I have taken a lot of question. I collected a lot of question, NEET as well as other entrance exam question covering whole topic. So we don't have time today to continue. Better we will be continuing on other day. We will be discussing remaining question up to this NEET 2026 exam. Okay, so we will be doing that. So better we will be continuing another day. Okay, so by saying this, I am finishing this part, and part 2 we will be continuing later. Okay, since time is limited. So that's it, guys. So we will be we will see in next class. Whatever your experience, just let me know through comment section. And thank you for joining. Okay, all the best.